Welcome back, everybody. I hope you had time to refresh yourselves. So this morning we had a very intense session where we discussed the design and the analysis of clinical trials. And I'm sure you all noticed that the emphasis was very much on evaluating the efficacy of drugs, determining whether they had the effect they were intended to have. We did not talk about safety, and this brings us to our second session. Obviously, safety of the drug products that come through our purview is a fundamental issue, a fundamental responsibility of FDA. Safety considerations change as we move through the drug development process. Early in the drug development process, we're concerned about the safety of trial participants. Later on, obviously, the concern becomes that of the general population exposed to the drug. The other thing that we didn't talk about this morning is that different groups in the population may respond differently to drugs. And that's another area which we're going to touch on in this session, which is special populations or subpopulations that need special consideration. So I'm delighted to welcome our speakers for this afternoon session. Shabnam Naseer, who will talk about the safety considerations in clinical drug development, and she'll be followed by Lynne Yao, who will be talking about specific populations in clinical trials. So at this point, let me hand it over to Shabnam.
Hi, good afternoon, everyone. My name is Shabnam Naseer. As was stated, thank you for the kind introduction. I'm a lead physician in the Division of Anti-Infectives in the Office of Infectious Diseases. And my talk today will be focused on safety considerations in clinical drug development. I do appreciate having the opportunity to speak today. So our learning objectives include the following. We'd like to understand that protection of subjects should always be the first priority when designing early clinical studies. We'd like to learn key safety considerations in the conduct of phase 1 trials, and to understand the principles of safety monitoring and reporting in clinical trials. So the main objectives of phase 1 trials are to assess safety and tolerability of the drug product, attempt to characterize dose-limiting adverse reactions, determine maximum dose associated with an acceptable safety profile, to characterize pharmacokinetic parameters, and explore drug metabolism and drug interactions. The subjects in phase 1 trials generally tend to be healthy volunteers, and these volunteers have few confounding factors. In some instances, phase 1 trials are conducted in patients, and these are generally when the drug is known or expected to be toxic. An example would be a trial that's evaluating a cytotoxic agent. When patients are enrolled in phase 1 trials, there will be more confounding factors and sometimes more difficulty in trying to separate disease-related manifestations from adverse reactions. In addition, special populations such as patients with renal or hepatic impairment can also be enrolled in phase 1 trials. So here are some general considerations when one is moving from the non-clinical to first-in-human study. So we have to consider what is the evidence we've garnered so far from the non-clinical studies. And so we have to ask ourselves a number of questions. What is the duration and the total exposure proposed in humans? And how does that correlate with what was tested in the animals? We have to think about the characteristics of the test drug. Is it a small molecule? Is it a biologic? Does it have a long half-life? We have to consider the disease that's being targeted for treatment, because that would need to be taken into consideration in the benefit and risk assessment. We have to think about the population in which the drug will be used. Is the intent to enroll women of childbearing potential or children in the study? And what's the route of administration? Is the product being given systemically or topically? So these are some of the initial considerations. Do the non-clinical studies that have been done so far provide sufficient data to allow for the proposed clinical trial to be undertaken? So what species were they studied in? Are the species appropriate and are they relevant to the particular drug? What are the potential target organs of toxicity? What was the duration studied in animals, and what was the dose and the route of exposure? Was it appropriate and similar to what is being proposed in the human study? And what about the PK and PD assessments? Were they appropriate? Was a dose response identified? And then again, do we need to do any special studies in animals if we're planning to enroll, for example, children or pregnant women? So it's important to keep in mind that only some toxicities that are seen in non-clinical studies translate into adverse reactions in humans, while there's other toxicities that do not. Excuse me. And so you end up with both predictable and unpredictable toxicities, and these can appear in any phase of development, and sometimes you actually only see them in the post-marketing setting. It really depends on the size of your safety database that was available pre-market. It's also important to keep in mind that certain adverse reactions are subjective, and these can be adverse reactions such as headache or other events such as hypersensitivity reactions that really can't be assessed in the non-clinical arena. So let's talk about some examples. So here's an example of predictable toxicity. And this example is from the anti-infective space where I'm from. I'm sure you can find other examples in other therapeutic areas. So linezolid is an antibacterial drug that was approved in the late 1990s, and at that time it was the first member of the oxalidenone class. It had activity against gram-positive organisms and was also thought to have activity against some resistant organisms. So certainly there was some therapeutic benefit that could be offered by the product. The non-clinical studies that had been conducted showed that myelosuppression occurred, and this appeared to be both dose and time dependent. There were various changes in the bone marrow, and it appeared to affect all three cell lines. But the product had the potential to offer therapeutic benefit, and so further clinical development was pursued. When phase 3 trials were conducted, there was no evidence of myelosuppression per se, but there was a high frequency of thrombocytopenia that was seen in the linezolid-treated patients. And at the time the product was approved, the prescribing information included precautions for thrombocytopenia, and they also noted a clinical the animal pharmacology section described the hematopoietic effect noted in animals. After the product was approved, there were cases of myelosuppression seen affecting all three cell lines, and these were reported as leukopenia, anemia, etc. And so labeling was updated to include a warning regarding myelosuppression. Here's an example of unpredictable toxicity. So we had two products, both were members of the beta-lactam class, which is a very well-known class of antibacterial drugs, and we have a lot of experience in this class. The structure of these beta-lactams, however, had been modified to enhance the spectrum of activity. There were no unexpected toxicities seen in animals, so both of the sponsors proceeded to conduct their phase 1 studies. The single dose went well, but then when they went into the multiple dose trial, the subjects developed moderate to severe skin reactions, such that the product development actually had to be halted for both products. So in this case, this is an example of unpredictable toxicity, and even if the non-clinical studies appear benign and you don't see any adverse events, it doesn't mean that you're not going to see events when you conduct the study in humans. Now to a discussion on maximum recommended starting dose. And you will be hearing more about this in day 2 lectures, but I'll just go over some high-level concepts from the clinical perspective. So there are some key principles in selecting an MRSD. One is that you want to avoid toxicity at the initial clinical dose, but at the same time you want to allow reasonably rapid attainment of the trial objectives, and these are usually tolerability and PK, so that you're not so low that it'll take you forever to get where you need to be, where the target dose was. And typically we use an algorithmic approach based on what doses were given to animals and what toxicities were observed. There are some alternate approaches based on animal pharmacokinetics and modeling, but for the most part we use the algorithmic approach. Some of the key concepts when you look at the MRSD are the no observed adverse effect level, or the NOEL, and this is the highest dose tested in animal species that did not produce a significant increase in adverse effects compared to a control group. The second term is HED, or human equivalent dose, and this is really what is the dose in animals that converts to a certain milligram per kilogram dose in humans. Based on that, you would be able to come up with the starting dose. Then you also have to consider which animal species you use to make these calculations. Ideally, you would choose the most sensitive species, and that's the species from which you can identify the lowest HED. In some cases, however, you would want to use the most appropriate animal species because that species actually might have the particular target of interest, and this is especially seen with biologics. So here's a schematic that shows the steps in determining the maximum recommended starting dose, and for your reference, the guidance is at the bottom of the slide. So first you would determine the NOEL, which is a starting point for determining a safe starting dose for new molecular entities in healthy volunteers. Then that gets converted into the HED. You select the HED from the most sensitive or appropriate species, and then you choose a safety factor. Then you divide the HED by that safety factor to come up with a maximum recommended starting dose. So what is the safety factor? It's a number that gives you a margin of safety for protection of human subjects when you're moving from animal studies to humans receiving the initial dose. The default number is 10, but there is some degree of flexibility, and sometimes we can go higher or lower than 10. The safety factor provides us some reassurance that when we're moving from animals to humans, that we're not exposing human subjects to any great risk. One knows that there's variability when you move from animals to humans, and the safety factor allows for that variability to be accounted for. So, as I mentioned previously in the examples, there are uncertainties when you go from animals to humans. There can be differences in the sensitivity between animals and humans to certain products. There can be difficulty in detecting certain toxicities in animals, such as headache, myalgia. There could be differences in receptor densities or affinities. You always have to be prepared for unexpected toxicities. In addition, there could be interspecies differences in how the product is handled, such as ADME. And keep in mind that, again, humans and animals may not always handle the product in an identical manner. So when would we have to increase the safety factor greater than 10? If it's a brand new class and we have no prior experience with the product, you might have to consider increasing the safety factor. Other issues could be that the toxicities in the non-clinical studies were severe or irreversible, or they could be toxicities that are non-monitorables. For instance, if you have histopathologic changes, but they were not monitored because there's no clinical methods to monitor those toxicities, or there are no biomarkers, that could be a reason to consider increasing safety factor. If there's a very steep dose response curve, that could indicate a greater risk in humans. If the PK is not linear, it limits the ability to predict dose-related toxicity. Also, if there's variable bioavailability, because you could have poor bioavailability in animals, and that may underestimate the toxicity when you move to humans. On the other hand, you could decrease the safety factor to less than 10, and some of the reasons would be if you're dealing with a member of a well-characterized class of drugs, if the toxicity is easily, that's produced is easily monitored, if it's reversible, if it's predictable, and also if the NOEL was determined based on toxicity studies of a longer duration, you might have a little bit of more reassurance in reducing the safety factor. So let's discuss an example of how you would calculate the MRSD. Here's a product for which the HED was about 400, derived from rats. The sponsor proposed a starting dose of 100 milligrams, so that brought the safety factor to 4. And the rationale provided was that this was a member of a well-characterized class of drugs, and that the toxicity studies in 2 species were of longer duration than what was proposed in the clinical trials. In addition, the potential toxicities were readily monitorable and reversible. However, we actually didn't agree totally with the rationale because we knew that members of the class had exhibited more toxicity than the parent class from which it was derived. And there was also some concern that the bioavailability in animals was low and that human bioavailability could potentially be greater than anticipated, leading to a risk of higher exposure in humans. So we actually agreed upon a starting dose of 50 milligrams, raising the safety factor to 8. So now let's move into some general considerations when you're going to be conducting these phase 1 trials. So more questions to ask yourself when you're getting ready to conduct these trials. One is, are the clinical trial protocols designed appropriately to ensure safety and meet the stated objectives? You have to protect the safety of the subjects in the trial as always. And do you have enough information about the quality of the investigational products? For example, has the formulation been well characterized? Do we know, for example, what the impurities are? Are the route and the rate of administration appropriate? Can the product be given as a bolus dose, or are there toxicities such that we would have to give it as a slow infusion? What is the mechanism or mode of action? Is it novel? Do we know anything about similar products and that act via a similar mechanism? Are you expecting an off-target effect? And that's a very important concept, especially if you're dealing with a biologic and the mode of action involves a target which is connected to multiple signaling pathways or affects biological cascade or cytokine release. Let's go into safety considerations regarding dosing. How do we determine an appropriate dose? Ideally, one subject should receive a single dose, and that would be followed by sequential administration within the cohort. You have to ensure that there is an adequate period of observation between dosing so that one has the time to observe and interpret adverse reactions in the cohorts at the lower dose. And how long you actually observe will depend on the properties of the drugs, the PK properties, how long it stays in the system, how long its half-life is. And so we also learn good lessons from previous trials conducted with similar products. So moving on to dose escalation. If the protocol for these phase 1 trials allows for dose escalation, you have to ask whether the dose escalation scheme is appropriate. Are the dose increments appropriate? Is it okay to double up or go up by 4 times? You have to be cautious and use a slower rate of dose escalation, especially if you have a small therapeutic window, if you had animal models that were not very good, or if you had any concerns about toxicity. There is a fair bit of flexibility here. Again, it just depends on your product, the target, and the toxicity found in animals. You have to ask yourself if the amount of information before each dose escalation is appropriate, and also if the number of subjects at each dose is appropriate. So next we'll move to safety monitoring. It's important that protocols have an appropriate monitoring scheme to look at clinical signs and symptoms of adverse events likely to be associated with the drug. And the duration of the clinical observation should also be adequate with respect to the stated objectives and endpoints, the anticipated response to the product, and the health-related conditions that are being studied. Sometimes we do need to monitor for a longer period of time, and again, it depends on the characteristics of the drug. Where we monitor also matters, whether it's a hospital setting that's needed. And if you're worried about any sort of delayed onset reactions, undetected toxicities, you have to make sure that the follow-up is long enough. So as far as frequency of monitoring, there may be a need to monitor more frequently soon after initial dosing in the first week or so. Certainly you would need to have follow-up visits for subjects who develop adverse events or laboratory abnormalities. In addition to monitoring clinically for adverse reactions, you'd also monitor laboratory tests. And then which particular laboratory tests you order and collect the data on really depends again on your drug and the target organs of toxicity. You want to ask yourself if the laboratory tests would include routine assessment of all organ systems. Do we know enough about the drug such that you only need to assess specific organ systems? Are they sufficiently detailed laboratory tests that you can actually make an assessment of whether or not the drug is causing toxicity of those specific organs? And then lastly, are there stopping rules for patients whose laboratory test abnormalities reach a certain threshold? So what are stopping rules? It's important that the protocol have clear stopping rules for administering the drug, enrollment, dose escalation, and rules for termination of the trial itself. Oftentimes, we've actually had to put INDs on hold because there were no stopping rules in the protocol. So again, these are changes to the protocol that are to be implemented when toxicity is observed. And to be able to have stopping rules, you have to identify what might be acceptable toxicities and what are not acceptable toxicities. Since it's unlikely that you'll have a drug with no toxicities, it's important to stop and try to generate these stopping rules. If acceptable toxicities occur, these will be the ones that don't result in changes in how the trial is continued in terms of subject enrollment and dosing. If you have other toxicities that are not acceptable, you have to have a procedure in place for what you would do with the study. So let's go on to our challenge question 1. Which of the following is used to calculate the MRSD? And hopefully everybody will get this. And this is the Noel. All right, so let's move on to sort of the second half of my talk, where we talk about where we continue to talk about the evaluation of safety, but sort of in larger, later clinical settings. So safety evaluation is a central component in all stages of drug development. It's an evolving process. It depends on the stage of the product under development. For products that are marketed, the safety information is reflected in the product labeling. It's also called the prescribing information, or PI. While up-to-date safety information for products that are under investigation would be contained in the Investigator brochure, or IB. So again, these would be non-clinical findings from in vitro or animal studies, clinical pharmacokinetic safety data. And for unapproved products, the investigator's brochure is equivalent to the prescribing information. So what are the sources of safety information found in the investigator brochure or the prescribing information? Non-clinical data again can include in vitro or animal data, as well as information about the structure of the product and the manufacturing process. Clinical pharmacological data can include information about PK/PD And then safety data can come from clinical safety information from healthy volunteers and patients. Perhaps the most pertinent information, of course, will be the data from the clinical trials for the same indication, as these data are generally controlled for the patient population characteristics and the underlying pathological process, and also from trials for different indications. You can also have information in the IB from post-marketing experience and, IB or prescribing information from post-marketing experience, the published medical literature, and last but not least, safety profiles of drugs in the same class. So now, why is safety monitoring required in all clinical trials? The goal of safety monitoring in clinical trials is to identify, evaluate, minimize, and appropriately manage risk. And this will lead to our discussion of adverse event ascertainment and monitoring. So we have a number of regulatory definitions that we'd like to go over, and I have given you the CFRs for your reference. Adverse event is any untoward medical occurrence associated with the use of a drug in humans, whether or not it is considered to be drug-related. This could be an unfavorable sign, symptom, disease, abnormal lab, a vital sign, or other testing, or a worsening of the above or a constellation of the above. Adverse events can also be referred to as adverse experience. Some examples of ways that adverse events can be ascertained include spontaneously reported or observed signs and symptoms. Symptoms are signs that are reported as a result of a probe, such as a questionnaire or a checklist. And then testing of vital signs, laboratory studies, and special safety assessments can also be conducted, and these can include, for example, visual, hearing tests, or EKGs. Upon reporting, adverse events are characterized according to their severity, and there are a number of severity grading scales or tables that are available to provide guidance to sponsors or investigators for safety monitoring in clinical trials. These are optional tools. They are specific to the study population that's being evaluated, for example, patients or healthy volunteers. Clinical abnormalities may be represented as either mild, moderate, severe, potentially life-threatening, for example, in healthy volunteers. If you have oncology patients, AEs could be graded by a scale of 1 through 5, where 1 is mild symptoms to grade 5, which is death related to an AE. So again, they're specific to the study population. And it's important to keep in mind also that here are some examples. There's probably more. So again, it's important to keep in mind that the term severe is not the same as serious. What happens when an AE report gets to the sponsor? So the process includes conversion from the investigator's verbatim terms to the standard preferred terms using a specialized dictionary called MEDRA. MEDRA is the Medical Dictionary for Regulatory Activities, and it can be used to sort AEs and group-like events. Terms are used to calculate the incidence of AE. Here's a schematic to illustrate the MEDRA hierarchy. It starts with the highest level of terminology, and that's the least specific, which is the systemic organ class, or SOC. And you move down all the way to the lowest level terms. This is in order of increasing specificity. The PT represents a single specific medical concept. So clinical investigators play a really crucial role in the safety reporting, and you would want them to use the most scientific and consistent terminology when reporting adverse events, as coding problems could result in splitting or lumping adverse events and therefore missing safety signals during the safety data analysis. So splitting similar terms such as hypertension, high blood pressure, these can result in a lower incidence, or they could minimize or mask a safety signal. And on the other hand, lumping dissimilar terms to the same PT could obscure a safety signal under a lumped term. So for example, facial edema is very different from leg edema as far as pathophysiology, and a coding issue could be lumping all edema to the PT edema. In this third bullet, there could be a lack of an adequate term or definition to represent a syndrome of interest such as drug hypersensitivity or serotonin syndrome. And in this case, you may need a prospective case definition if the syndrome is not well characterized by a single term. Moving on to another regulatory definition of serious, again, not to be confused with severe. Serious adverse events are any adverse events that, in the opinion of the investigator or sponsor, results in the following. So death or life-threatening conditions with the immediate risk of death, in hospitalization or prolongation of existing hospitalization, a disability or a birth defect, and last but not least, important medical events. And these would be those that could jeopardize the patient or require intervention to prevent one of the outcomes listed above. Examples may be allergic bronchospasm, which would require intensive treatment in an ER or at home. So here's a list of examples of uncommon SAEs. And if you see one of these, it could raise a red flag that there might be a possibility that the AE was caused by the study drug. Especially if you have a study population of generally healthy individuals, you really shouldn't see one of these SAEs. This is not a comprehensive list, but it may be a good start. So on this slide, I'll discuss points to consider for investigators in evaluation of SAEs. SAEs are, like any other adverse event, need to be assessed for drug-related causality, and you can do that by asking a number of questions. One would be, is it common? Is it a common occurrence in the population under study? Was it treatment emergent, meaning did it occur on treatment or shortly thereafter? Did it respond to drug discontinuation or de-challenge? Did it reoccur upon introduction of the study drug or re-challenge? Were there concomitant medications? Were there any pertinent laboratory studies or other tests done to try to characterize the adverse event further? Was there an obvious alternative cause? And lastly, but not least, was the SAE a study endpoint? For example, if death occurred, was that also a study endpoint? So you also want to report adverse events by providing enough relevant clinical information in the case report form. You want to include enough information in the case report form to allow a good quality narrative. So now let's talk about what the clinical investigator reports to the sponsor. And again, the CFR is there for your reference. So an investigator must immediately report all SAEs, whether or not the event is considered drug-related. The investigator must also include an assessment of whether there's a reasonable possibility that the drug caused the event, and this is usually called a causality assessment. Study endpoints that are SAEs must be reported in accordance with the protocol. One exception is that if there's evidence suggesting a causal relationship between the drug and the event, then in that case, the investigator must immediately report the SAE to the sponsor. In addition, the investigators should also record non-serious SAEs and report them to the sponsor according to the timetable that's noted in the protocol. So let's discuss a hypothetical case. I think that this might illustrate some of the points mentioned on the last couple of slides. This is case 1. You're an investigator for a clinical trial evaluating whether antihypertensive drug A is associated with a reduced risk of death, MI, or stroke, and death is a study endpoint in the trial. You're told that an 80-year-old white male just died in the trial, and we're also actually told the cause of death was anaphylaxis. So the question is, do you have to immediately report this case to the sponsor? So let's take a minute to think about what to do here. So you're told that death is a study endpoint, and usually an SAE that is also a study endpoint can just be reported per protocol. However, here we're told that the death was due to anaphylaxis. So if you recall in the previous slides on uncommon SAEs, anaphylaxis was listed, and with that SAE, that should be sort of a red flag that there could be evidence to suggest a causal relationship between drug A and event, and so the investigator must immediately report this event to the sponsor. So the question is yes. The answer is yes to this question. All right, so moving back to some regulatory definitions, we'll discuss the ones important to expedited reporting of adverse events to FDA. And the concept of unexpected adverse event is presented here. An adverse event is deemed unexpected if it's not listed in the investigator brochure or IB, or if the IB is not available or required, if it's not consistent with the risk information described in the general investigational plan or elsewhere in the current application. It would be unexpected if it's listed in the IB, but it's not at the specificity or the severity observed. For example, the IB lists increased creatinine, but you have a case of acute renal failure, and that would be considered unexpected by virtue of greater severity. Other examples would be if you have a cerebral thromboembolism, and this would be considered unexpected by virtue of greater specificity if the IB listed only CVA or cerebrovascular accident. And it could also be considered unexpected if it's mentioned in the IB as anticipated due to the pharmacological properties of the drug, or if it occurs with other drugs in the class, but not with the particular drug under investigation. So one example would be angioedema. It's anticipated to occur in patients that are exposed to the class of ACE inhibitor drugs. Angioedema would be described in the IB as a class effect, but a case of angioedema observed with the drug under investigation would still be considered unexpected for reporting purposes until it's included in the IB as occurring with the drug under investigation. Go on to another hypothetical case, case 2. Here you're the clinical investigator for a trial evaluating a new anti-malarial drug B for the treatment of acute uncomplicated malaria. The investigator brochure lists elevated hepatic enzymes. And the question would an event of hepatic necrosis in this trial for drug B be considered an unexpected AE? So I'll give you a moment to think. So the answer is yes, hepatic necrosis in this hypothetical case would be considered unexpected. And the reason is because the IB does not list the AE at the severity observed. So you're seeing hepatic necrosis when only elevated hepatic enzymes are noted in the investigator's brochure. Okay, so moving on to our next definition, suspected adverse reactions. So a suspected adverse reaction is an adverse event for which there's a reasonable possibility that the drug caused the adverse event. Again, reasonable possibility means that there's evidence to suggest a causal relationship between the drug and the AE for purposes of IND safety reporting. So let's look at some examples of suspected adverse reaction. This could be a single occurrence of an uncommon event that is known to be strongly associated with drug exposure. So for example, angioedema, Stevens-Johnson syndrome. Another example would be one or more occurrence of an event that's not commonly associated with drug exposure, but is otherwise uncommon in the exposed population. So if you have healthy subjects and they're developing neutropenia, that could be an example of this, or a heart valve lesion in a young adult, or tendon rupture is another example. And another type of example is an aggregate analysis of specific events observed in a clinical trial, indicating that those events occur more frequently in the drug treatment group than in a concurrent or historical control group. And an example of this would maybe having several acute myocardial infarctions in long duration trials of elderly cancer patients. So overall, the determination of an AE as a suspected adverse reaction it can be difficult, but it's really critical to avoid submission of uninformative safety reports. And the sponsor is the one that should evaluate all available information and decide whether there's a reasonable possibility that the drug caused the AE. And that's because the sponsor is actually the ultimate party that determines if a drug is or is not causally related to the AE. Here I wanted to show a slide that illustrates the universe of suspected adverse reactions. So these are actually not just adverse events with undetermined causality, and adverse events here are noted in the outermost yellow sphere, but they're not yet definitely established adverse reactions, which are noted in the dark red sphere in the center. So suspected adverse reactions are actually in the middle. Adverse reactions are a subset of all suspected adverse reactions where there's a reason to conclude that the drug caused the event. Now let's talk about IND safety reporting by the sponsor to the FDA. FDA continues to require that 3 criteria generally be met before a sponsor submits an IND safety report. And again, the CFR is provided here for your reference. So namely, the event must be serious, unexpected, and there must be evidence to suggest a causal relationship between the drug and the event. Often this standard is called a SUSAR, and it stands for Serious and Unexpected Suspected Adverse Reaction. So again, the sponsor must assess whether or not there's a reasonable possibility that the drug caused the event, and the sponsor should only submit these type of IND safety reports when all 3 criteria are met. In addition, sponsors are expected to submit an annual report in the IND that includes a summary of the most frequent adverse events. It should also contain a summary of the SAEs. So now let's talk about timeframes when it comes to IND safety reports. These reports are required to be submitted to FDA in 15 days, and those that are required to be submitted within 7 days. As you can imagine, the most serious events must be reported within 7 days. And there are 5 categories of adverse events that trigger the sponsor's responsibility to submit an IND safety report. One is the SUSAR that we just mentioned, serious and unexpected suspected adverse reactions. Findings from other studies suggesting a significant risk in humans. Findings from animal and in vitro testing suggesting a significant risk in humans. An increased rate of occurrence of serious suspected adverse reactions, and serious adverse events from bioavailability and bioequivalence studies not under IND. And sponsors have to submit IND safety reports within 15 days of the adverse reaction unless the adverse reaction was fatal or life-threatening, in which case the report must be submitted to FDA within 7 days. And there may be additional details that you might want to look at in the guidance that I've provided in my references regarding the timeframes. So in summary, I've tried to provide a high-level overview of safety considerations in phase 1 trials, important questions to ask yourself before you decide to dose humans as you translate the information from non-clinical studies to either healthy volunteers or patients. what's the relevance of toxicities seen in the non-clinical studies, and how they translate to adverse events in humans. It's important to keep in mind that there are both predictable and unpredictable adverse reactions. We also discussed how to go about determining a safe starting dose in humans and the concept of the safety factor. We next discussed the importance of safety monitoring and stopping rules, and the ascertainment of safety in larger clinical trials, and some important regulatory definitions of adverse events and monitoring. Finally, we also discussed how investigators play a really integral role in assuring quality safety assessments and the requirements of reporting to the sponsor. And then in turn, we discussed about how the sponsor will evaluate all available safety information and report to the FDA and all participating investigators. Okay, so this brings us to our last challenge question. This is a true or false question. If the adverse event meets all three of the definitions for suspected adverse reaction, serious and unexpected, it should be submitted as an IND safety report. So I'm going to give you a few seconds to think about it. So the answer here is true. I hope everyone got this one. All three criteria should be met for it to be submitted as an IND safety report. Now I just wanted to show you my references. If you had any more questions, please refer to these. And I thank you so much for your time today. Appreciate the opportunity to speak. And the next speaker will be Dr. Lynne Yao, who will present on special populations. Thank you very much.
Hello everyone, I'm delighted to be here today to give you some of my perspectives on specific populations in clinical trials. Here are the objectives of my talk. I'll be focusing on drug development considerations for children and pregnant and lactating individuals, and I'll mention some of the work we're doing currently in enhancing diversity in clinical trials. So FDA labeling regulation includes requirements for labeling of specific populations, and you can see here some of the specific populations that I'll be talking about today. There are other specific populations that I will not be discussing, including geriatric populations and patients with renal or hepatic impairment. So let me start this section with describing the historical problem for children. Prior to about the late 1990s, very few drugs that were used in children were actually approved for use in children, which of course led to significant off-label use. Why was this the case? Well, first of all, we understand clearly that there are differences between children with respect to development, drug metabolism, and toxicity, potential toxicity issues that require that we study drugs in children if we're going to use them in children. But often those studies were discouraged because of many issues, including ethical issues, fear of harming children, and of course perceived liability. There was also a lack of incentive for drug companies to conduct studies. And what this all led to was a situation in which practitioners were faced with a choice, either to not treat children with potentially beneficial medicines because they were not approved for use, or to treat patients based on limited data, anecdotal experience, or based off information from adult studies. In the late 1990s and early 2000s, there became a clear recognition that children should have access to products that have been appropriately evaluated, and that when drug developers are developing drugs for an adult use, if that drug would have a potential for use in children, that those product development programs should include pediatric studies. And of course, there's an important regulatory standard that must be met for conducting pediatric research. And these regulatory standards increase the strength and the quality of the research. These concepts were all outlined in a very important document, the ICH, that's the International Conference on Harmonization, E11 and E11R1, clinical investigations of medicinal products in the pediatric population. Around the same time that the ICH E11 was originally published in 2000, there was a movement in the United States to ensure that children had access to approved drugs as well. And so there were advocates, children advocates across the country who lobbied Congress to pass pediatric drug development laws. And what we had passed in 2002 was the Best Pharmaceuticals for Children Act, or BPCA, and then shortly following in 2003, the Pediatric Research Equity Act, or PREA. PREA, as you can see on the left-hand side, requires studies. So FDA is given the authority to require studies under certain circumstances for diseases or conditions that occur both in adult and pediatric populations. Now, importantly, products with orphan designation are exempt from requirements under PREA with the exception of certain pediatric cancer drugs, and information from those studies that are required by FDA must be labeled. On the other side, BPCA is a voluntary or incentive provision, which allows for 6 months of additional marketing exclusivity if studies are completed and submitted to FDA that are in response to FDA's request for certain studies. Now, it's also important that orphan designated products can be included under BPCA, and information from those studies must also be included in labeling. Now, you can see that there's an intersection between PREA and BPCA, and we believe that these two laws work together hand in hand to increase the availability of approved drugs and biological products for the treatment of children. So on the next few slides, I want to provide some comments on therapeutics development in children. And importantly, in the US, there's an evidentiary standard for approval, and that approval is no different for children compared to adults. And that is, as you've probably heard from previous talks, that in order for a drug to be approved in the US, there must be demonstration of substantial evidence of effectiveness and clinical benefit. That is, how a treatment impacts a patient's ability to feel, function, or survive, or that it improves or delay in the progression of clinically meaningful aspects of a disease. And that evidence is usually required through the conduct of adequate and well-controlled investigations. And there must also be important safety information included to apply an appropriate risk-benefit analysis. So while the approval standards are the same for children and adults for drugs in the US, there are special considerations that relate to children. First, and very importantly, are the ethical considerations. And I could give a talk for an hour just on the ethical considerations. But the 3 points highlighted here are very important and include that children should only be enrolled in clinical trials if the objectives and the scientific data necessary cannot be met by enrolling a population that can provide informed consent. Also, a child cannot be enrolled in a clinical trial unless there's a prospect of direct benefit. And that's important for any trial that would expose a child to a risk that would be considered any more than a minor increase over minimal risk. And then also, children should not be obviously placed in a disadvantage for enrolling in a clinical trial. There's also important feasibility considerations with respect to pediatric product development. And this is, I guess, a good thing for children, and that is that generally children are healthier and that the prevalence or incidence of disease in many pediatric populations is lower than that seen in that same disease in adults. Nevertheless, these feasibility considerations can play an important role in successful completion and the design of pediatric clinical trials. So I've described the ethical and feasibility differences in therapeutics development between children and adults. There are additional special considerations that I want to go over on this slide. Firstly, formulation development may be different, and that may seem obvious to many of you, but children below a certain age may not be able to take and swallow safely in a solid oral dosage form, so there may be a need for different types of formulations that are acceptable to children. Additionally, even if you have an appropriate oral dosage form, you may need to consider different strengths or different concentrations to allow for accurate dosing of all pediatric age groups. Animal toxicology studies, there may be a need when there are specific developmental safety concerns to conduct specific juvenile toxicology studies, and I provided a very important guidance, and another ICH guidance, S11, that you can refer to when considering the need for juvenile toxicology studies. There are clearly study design considerations ranging from dose selection to the choice of endpoint or control, as well as the monitoring of safety, including developmental safety issues that should be considered. And then the timing of the studies. When should the pediatric studies be initiated relative to the initiation or the completion of adult drug development? And then finally, and I'll talk later on about the use of pediatric extrapolation, a very important framework to consider, and I'll speak about that in a few minutes. Many of these, or all of these considerations, are discussed, as I mentioned, in that ICH E11 guideline, and then the revision to the guideline, and I've created the link here for you. So now I want to discuss pediatric extrapolation. As I mentioned briefly on the previous slide, this is a very important concept that can be applied specifically in pediatric therapeutics development programs. This new guideline that was published in August of this year, the ICH E11A Pediatric Guideline, goes into very important details on how to apply pediatric extrapolation. But let me just spend a little bit of time to explain how that can be applied in a pediatric development program. First of all, pediatric extrapolation is defined here in the first bullet. But what I really want to explain to you is that in an adult development program, in a successful one, when you've established efficacy and safety, in the subsequent pediatric development program for that same condition, if it is determined that there is sufficient similarity between the disease, the drug pharmacology, and the response to treatment between the adult reference population and the target pediatric population, then you may be able to develop or design a program that can leverage the data already collected on safety and efficacy in the adult program and apply that to the pediatric program. Clearly, if data can be leveraged in an adult program that would support a pediatric program, this could lead to an increase in the efficiency and success of pediatric drug development and greatly decrease the time necessary. However, in order to apply pediatric extrapolation, as described in the guidance, there needs to be robust information to support the similarities between the adult and pediatric populations. And importantly, any differences that may affect the degree to which pediatric extrapolation approaches can be applied. The guidance also describes that efficacy and safety can be extrapolated given the appropriate support and justification. And another important point about developing a pediatric extrapolation approach is the need to have multidisciplinary expertise, including specialists in pediatrics and in the specific disease, clinical pharmacology experts, as well as biostatistical expertise. There are many examples of pediatric extrapolation that I could go into, which I won't, because I think it's better to refer you to this guidance and to read it carefully, because there are great examples within the guidance, as well as training material examples at the ICH website that I can direct you to for more information. So now that I've given you a taste or flavor of the important components of a pediatric drug development plan, here's where I want to talk to you about some regulatory requirements. So under PREA, as I mentioned, the Pediatric Research Equity Act, there's a requirement for sponsors to submit their plans for how they're going to study children that they may be required to study under PREA in the submission to FDA of an initial pediatric study plan, or IPSP. This guidance was finalized in 2020. And in this guidance, it goes through important considerations for what to submit, how to submit, and when to submit these IPSPs to FDA. And this IPSP is really intended to provide FDA with a chance to review plans for pediatric therapeutics development and to provide advice and recommendations about those plans. Importantly, you can get a lot of information from the guidance, and I've provided the link here. But we want these plans submitted as early as possible during drug development, but certainly no later than the end of phase 2 in most situations. So what have been the effects of BPCA and PREA over the last almost 25 years now? And as you can see here, again, prior to the late 1990s, almost no drugs had pediatric-specific labeling. And you can see here that there's been really a clear linear increase in the number of labeling changes with pediatric-specific information over the last 20 years. And I'm happy to report that we have over 1,100 labelings with pediatric-specific information over the last 20 years. So I believe that this is evidence of the effect, the very important positive effect that the laws in the U.S. have led to drugs that are now appropriately labeled for use in children. Now I'd like to turn my attention to inclusion of pregnant individuals in clinical trials. Some important background information. There are approximately 5.5 million pregnancies in the United States each year, and clearly pregnant women may have chronic conditions that exist before they're pregnant, or they develop conditions while they're pregnant, and they can also have conditions specifically related to their pregnancy. Just like children, though, pregnant individuals have historically been excluded from drug development trials, and oftentimes at the time of approval, the only safety information we have come from non-clinical, usually animal reproductive toxicology safety studies. So here on this slide, I want to make some remarks about similarities and differences between adults and children, specifically pregnant and lactating adults. So children, pregnant and lactating adults should have access to medicines that have been appropriately evaluated. But when a drug is approved in adults, it's also approved in pregnant and lactating adults, which is unlike the situation in children, where approval in adults does not automatically mean that safety and effectiveness have been established in children. However, in pregnancy and lactation, there are important physiological changes that can impact the safety and dosing of drugs. And oftentimes at the time of approval, the only information we have are from animals. Therefore, there's an important need to consider collection of safety and dosing information in addition to what has already been collected in non-clinical studies. And this is all to support prescribers and patients being able to make informed decisions about whether to take a drug during pregnancy or lactation. As I mentioned, just like children, there has been this general approach to automatically exclude pregnant or lactating individuals from clinical trials. But there's been an increasing awareness over the last few years that there needs to be a paradigm shift, and that there are interested stakeholders and advocates that recognize that pregnant and lactating individuals can be safely included in clinical trials under certain circumstances. There are also regulatory advances that are occurring to change this automatic exclusion paradigm, including that the common rule, which is a regulation that is promulgated by NIH to remove the reference of pregnant individuals as vulnerable, and FDA is working to harmonize this regulation with the NIH regulation. So what are some issues that must be considered before including pregnant individuals in clinical trials? I want to point out that FDA published a guidance on this topic. It's a draft guidance in 2017, and this information is all contained in the guidance. But let me go over some important highlights. The first is, before pregnant individuals can be studied, there must be at least some margin of safety established through the conduct of non-clinical studies in pregnant animals. And that's no different than going into adult populations or pediatric populations, that we must have the safety identified or evaluated, generally first in non-clinical studies. In the pre-marketing setting, it's important, again, because there is the need to understand the benefit to the pregnant woman and fetus before enrolling a pregnant individual in a clinical trial pre-marketing, that there must be a prospect of direct benefit, and that information that you obtain by enrolling that pregnant individual cannot be obtained by other means. In the post-marketing setting, generally we'd like to have some safety data available in non-pregnant individuals, adults, and that there may be some information to support it from the literature. Now, in addition to that two conditions exist. One, that efficacy can't be extrapolated, and two, that you can't get the safety information from any other means. And these are important principles that need to be present before enrolling pregnant individuals, either in the pre-market or post-market setting. The other thing that's important that's discussed in the guidance is what happens when a patient becomes pregnant during the clinical trial. And there are important considerations there, and to not lose the opportunity to collect some information because that patient became pregnant during the trial. Now, that patient should be immediately unblinded so we know whether or not the patient was exposed to drug or whether not through or just placebo, and that the patient should undergo a reconsenting process, because that patient may actually want to continue in the study because the exposure has already occurred, and there's the ability to collect important additional safety as well as PK and dosing information. So in addition to the guideline that FDA published in 2017 about inclusion of pregnant individuals in clinical trials, a report was recently published by the National Academies of Science, Engineering, and Medicine. And this report was specifically to evaluate liability associated with enrollment of pregnant individuals in clinical trials. And the report's key findings are listed here, and somewhat surprising. And again, I would be happy to discuss this offline with those of you who might be interested. But one important finding is that there's a lack of evidence of liability actually related to enrollment of pregnant and lactating individuals in clinical trials. But there is potential liability after approval if pregnant individuals are exposed to those drugs, liability to the prescribers and potentially to drug manufacturers. And the report also concludes that liability could actually be prevented by including pregnant individuals in clinical trials. And I've given you the link to the report. It's quite a detailed report. And again, there are some limitations in these findings, but I think the report really highlights some perceptions that maybe need to be changed about inclusion of pregnant individuals in clinical trials. I also want to highlight ongoing work at the ICH level, that there is a guideline under development right now on inclusion of pregnant and breastfeeding individuals in clinical trials. And you can see here what the goals of that harmonized global guideline will include. So in the last few minutes, I want to discuss what FDA is doing to enhance diversity in clinical trials. So on this slide is a quote from our current FDA Commissioner, Dr. Rob Califf. And basically, Dr. Califf has made ensuring diversity in clinical trials an important issue for FDA to tackle because it is a fundamental public health issue. Now, in addition to his quote, FDA has been working to ensure the health of the whole public, including underrepresented populations, and that clinical trials should reflect the population that is most likely to use the drug. And that includes populations who have generally been underrepresented in clinical trials. So I don't have time in detail to go over all of FDA's efforts to ensure diversity in clinical trials, but suffice to say, you can see that FDA has been very busy over the last few years with guidances and workshops to help sponsors understand and incorporate diversity in clinical trials. I will also want to especially point out at the bottom the ADEPT-9, or the Advancing Development of Pediatric Therapeutics. Our 9th conference was specifically convened to discuss enhancing diversity in pediatric clinical trials. And this was just convened a couple of months ago. There was very robust discussion, and patient advocates as well as patients themselves discussing the importance of diversity in clinical trials. Just a quick word about how decentralized trials may enhance the ability to enroll a diverse population. So decentralized clinical trial is just a trial where not all of the information is collected at the investigator's location, but some or all of the trial activities could be conducted at a remote location from the clinical trial sites. There are clear potential benefits, including patients not having to travel to those centers, take time off of work, find childcare, etc. That it can increase enrollment, particularly for rare sporadic diseases where patients may need to travel hundreds of miles to get to a site. And even when they are enrolled, it can improve continued participation and maintaining patients in clinical trials and avoiding dropouts, because some or all of those endpoints and measurements can be collected closer to the patient. Now, obviously, there are challenges related to that, including the need for training, as well as tracking strategies, because you're likely going to need more personnel in a decentralized trial. And then, of course, compliance issues that need to be worked out to ensure that the data collected are valid and reliable. So in summary, I hope to have conveyed to you that children, pregnant, and lactating individuals deserve to have access to medicines that have been developed with sufficient information to support their safe and effective use in these specific populations. That drug development should be designed early to include these specific populations and plans to collect data in these specific populations when it's expected that if the drug is approved, it will be used in these specific populations. Of course, that requires early planning. And just some, what I would say, catchphrases that I'd like to start hearing and I'm bringing and presenting to you right now. That the health of the child begins with the health of the mother. And that children, pregnant, and lactating individuals are protected through research, not from research. And finally, that the U.S. population has become increasingly diverse, and clinical trials should reflect the population most likely to use the drug. And so there's a need to enhance the diversity of the clinical trials we are conducting for the specific benefit of all patients who could benefit from the use of an approved drug. Thank you very much for your attention. And before I forget, I have a challenge question for you. Specific populations that should be considered for enrollment in clinical trials during drug development include A, pediatric populations, B, pregnant and lactating populations, D, all of the above, E, none of the above. And I'll give you a minute to answer. And the answer, of course, is D, all of the above. Thank you again for giving me this opportunity to present to you today.
Well, thank you very much for these great presentations, Shabnam and Lynne. I think you've really very comprehensively covered the topic of safety evaluation early and late in clinical trials. And Lynne, thanks for addressing really some of the most important subpopulations, which are obviously addressed in regulations and requirements as well. So I'm going to move on to questions, and there are many of them, excellent questions from the audience. I'm going to start off with Shabnam. Shabnam, there's a question for you. What role do investigators play in ensuring patient safety during clinical trials, and what measures are in place to monitor and report adverse events? So this is about the role of investigators in safety.
Okay, yeah, thanks, Leonard, for that question, and thank you for the audience as well. So what role do clinical investigators play? Yeah, that's a great question. Investigators play a key role, as you heard during the presentation, especially when it comes to reporting SAEs to the sponsor. Keeping in mind the exceptions that we discussed in the talk, investigators are involved in determining if the AE is serious, and you could refer to the list of questions to ask yourself about what constitutes the AE as serious. There were, serious is a regulatory definition, so it's clearly defined there. As investigators, you're required to provide a causality assessment for each of the SAEs reported to the sponsor, and that really helps the sponsor because the investigator's view is important when considering, when assessing the safety of the drug and determining whether to report the event eventually to FDA. It's the investigator who monitors the subject's response to the drug and is knowledgeable about the subject's clinical state. They know the medical history and the concomitant medications that the patient was on, and so they may be especially sensitive to distinctions between events that could be related to the drug versus those that might be due to the underlying disease process, for example. Over, Leonard. Thank you.
Thanks very much, Shabnam. I think obviously emphasizing very important points that probably nobody understands the adverse event as well as the investigator saw it. So obviously a very important source for information from us. So Lynne, I have a question for you. Interesting question which has come up here. If a trial protocol excludes pregnant women and a pregnancy occurs during a trial, what are the regulations or best practices for follow-up on the baby, including the length of follow-up? So what happens if somebody gets pregnant during a trial? Any thoughts there?
Thank you, Leonard, for that question, and thanks for the question being asked. So even when it's appropriate to exclude pregnant individuals from clinical trials, and I hope I've convinced you that we should move away from the automatic exclusion of pregnant individuals from clinical trials. But even when they are appropriately excluded, pregnancies happen. Not every contraceptive measure is 100% effective. And so I think the question is a very good one. Part of the importance is not to automatically remove the patient from the clinical trial, stop all follow-up, and lose that patient from potential important follow-up measures. So one of the things I think I mentioned earlier is the first thing that should happen is that the patient, if it's a blinded controlled trial, you have to break the blind on that patient because you need to know if that patient was exposed to the test treatment. After that, then what should occur is a question about what the risk benefit is to continuing that patient in the study, even if they are pregnant, and to reconsent them in that process. In other words, once you know if the patient's been treated, then they might be able to be continued in the study, at least for safety, even if we have to break the blind. But that requires the consent of the patient. So one of our best practices that we describe in the clinical ethics guidance that we have on pregnancy is to reconsent that patient and to continue to follow them on treatment if that's appropriate. And in addition, one thing that I think is very important is to collect PK data on that patient, because the PK data can help us understand better whether or not there needs to be dosing adjustments during pregnancy. Now, the second part of that question is what about the fetus and what about the infant after it's born? So again, depending on the situation, for example, with biological agents that are immune modulators, particularly if they're given in the third trimester, can pass and cross into the fetus through the placenta. And that can potentially have effects on the immune system of the newborn and infant. So depending on the drug that is under study, there might be a need to follow that infant, not just after birth, but for some period of time after birth, even and including up to a year or more. So we do have information about that in our pregnancy clinical ethics guidance related to pregnancy that I can direct you to. But it's a very good question. And there are a lot of things that can be done besides just saying that the pregnant individual has to be removed from the trial and not followed up. Thank you, Leonard.
Thanks very much, Lynn. I think an excellent explanation, emphasizing really the importance of getting the maximum amount of information about these situations that happen during the course of a clinical trial, both in pregnancy and obviously in exposed neonates. Shabnam, a question for you. Here, since an AE can include a worsening of an existing condition or may be new but appear gradually, when should the beginning of the AE be considered insofar as trial records are concerned? So I guess the question here is related to when do we regard the adverse event as beginning in relation to when the trial, the patient was put on the drug?
Okay, thanks, Leonard, for the question. So the adverse event definition, just recap that it's any untoward medical occurrence associated with the use of a drug in humans, whether or not considered drug-related. And adverse events that occur during the study drug treatment should be coded as treatment emergent to differentiate from events that may have occurred prior to study drug treatment. I think that's important concept. Treatment emergent adverse events, they're coded in MEDRA with a flag, usually called a treatment emergent flag, and that would distinguish them from other AEs. For example, perhaps one that may have occurred right prior to study treatment, perhaps a worsening of a condition even prior to receiving study treatment. If we're trying to distinguish AEs which may have been related to worsening of an existing condition, that did occur on drug, you may consider coding it in such a way that it reflects actual worsening of that event. So I hope that's clear. Over, Leonard.
Thanks very much. Okay, I have another one for you, Lynn. Would it be appropriate to recruit participants for studies to develop drugs for diseases that impact people from minority populations or children differently than drugs being developed for the general population? And the example that's given is this might be at hospitals, clinic, or disease support groups. So I guess a question targeted at drugs for diseases that impact people from minority populations. Any thoughts about that?
Yes, thanks, Leonard. An excellent question. One of the things that we have learned, particularly through that ADEPT 9 workshop that I mentioned in my talk, Enhancing Diversity in Pediatric Clinical Trials, I think it's right to state that, and as I mentioned in my talk, that, we need to study drugs in the populations that we believe are taking them. And so what we don't want to see necessarily is a drug development program for a disease that includes a substantial number of minority populations, that population is only studied in clinical trials that, for example, enroll a very non-diverse population. And I'll tell you why that's important. That's important because what we heard at this conference, this workshop that we convened, was that not only is it important to understand whether the drug behaves differently in a minority population compared to a non-minority population, but that when patients are recruited from distinct areas that have generally been underrepresented, that enrollment of those patients gives trust in those communities that this trial is worth something and this drug, if it's approved, is actually acceptable to take. We heard from many stakeholders, including patients, minority and underrepresented populations who said that, why would we take a drug that was approved by FDA because we don't trust the government, and nobody in our neighborhood even entered the clinical trial. But if there's enrollment of patients from those underserved areas and those patients go back and talk about their experience, not only does it help with recruitment in those areas, but it helps after, as I said, the drug is potentially approved and available, that it gives confidence and trust that those drugs are actually safe and effective, as FDA has determined that. So yes, I agree that it's important to enroll a diverse population, including those that might be historically underserved. And yes, those sites can include regional clinics. They can include engagement with disease support groups. And again, that idea of the decentralized trial is also one that can help with enrolling populations that have been historically underrepresented. Thank you.
Thanks, Lynn. I think you make an excellent point that inclusion improves trust in communities that are not generally participating, a really important consideration. Shabnam, I have a question for you. The question's simple. I think the answer has nuances. What kind of study will have death as an endpoint? Do you want to respond to that one?
Yeah, sure, Leonard. Thanks for the question. Yeah, what kind of study would have death as an endpoint? Death is an endpoint for several types of clinical trials, and especially for diseases that are considered serious and life-threatening, it can be very useful. An example of that would be assessment of an all-cause or a 14-day mortality, which can often be an efficacy endpoint. Again, in trials that are looking at conditions that are serious or life-threatening. And in the anti-infective world, one example could be a phase 3 trial for example, for a healthcare-acquired or healthcare-associated or ventilator-acquired bacterial pneumonia, in which we often have efficacy endpoints looking at mortality at a certain end time point to assess the non-inferiority of one antibacterial drug versus another standard of care antibacterial drug. And then in the talk, as you recall, the example was for an antihypertensive drug that was looking at endpoints related to mortality and acute myocardial infarction and stroke and that sort of thing.
Yeah, thanks very much. Sorry, Shabnam.
Yeah, that's it. Thank you.
Yeah, I just had some thoughts as well. Outside of the realm of infectious disease, obviously cancer trials are very often have mortality as an endpoint or time to death, because many of those patients do experience death. The other thing that probably is just that came to my mind was sometimes difficult to tell whether death is the endpoint as a result of a failure of the drug or whether it may be some other cause. And, I think sometimes evaluating death may be tricky, particularly if patients have serious underlying comorbidities. So I think it's a very insightful question. Let's see. I'd like to move on to another one for Lynn. This one's about dosage extrapolation, Lynn. The question says, we can't extrapolate dosage in neonatal populations because of the very different physiology related to gestational age, right? Any answers to that?
Yes, I do, Leonard. Thanks for that question. Again, another very insightful question. So it is true that, and again, in my discussion of extrapolation, it relies on how similar the population is with the disease, a particular pediatric one, compared to, for example, an older pediatric population or an adult population. And we certainly understand that neonates have a very rapid change in their physiology, their metabolism, their physical growth over a very short period of time. So it is hard to imagine that neonates could be similar to an adult population, for example. So we'd have to absolutely do separate neonatal trials. And the answer to that is that, it may be true that trying to compare a neonate to an adult would be difficult. But comparing a neonate to, for example, a 6-month-old or a 3-month-old, and if we have dosing information and efficacy established in those groups, it may be very reasonable not to have to conduct, a fully powered, randomized, placebo-controlled trial in the neonatal population, but leveraging what we know about that 2-month-old or 6-month-old population, that we would still need to collect probably safety information, and yes, to your point, dosing information, because we recognize that those metabolic enzymes develop rapidly during the neonatal period. So yes, we would likely need to have dosing information, and we would likely need to have safety information, but probably not necessarily in all cases separate neonatal randomized controlled trials. And the only other point I want to make too is that if, for example, this drug had been tested in neonates for a different condition, and now the same drug is being looked at for yet another indication in neonates, then having that information from another previous development program in neonates can be very helpful, and that information could potentially be leveraged. Thanks.
Thank you very much, Lynn. As a non-pediatrician, I can only imagine how difficult it is to study neonates for the activity of new drugs. Shabnam, I have another question for you. How is the term severe used in the classification of AEs?
Yeah, thank you for the question. I'm glad the question was asked because this does come up a lot. And so, I had mentioned in the talk that severe does not equate with serious, as that is a regulatory definition. But severity can be an indication of intensity of a specific event. So that's the key word, is the intensity when we're talking about how severe something is. Severity can be assessed by those toxicity grading scales that I had mentioned. And you can, reference some of those examples on the slide just to take a look at what may constitute mild, moderate, and severe for different patient populations. So in general, severe AEs would, as you would think, would be those that would cause significant pain, discomfort, for example, or may prevent daily activity, as an example, depending again on the study population. What may be severe for one patient population, like a healthy volunteer, may not be the same for oncology patients, for example. And then in terms of laboratory abnormalities, a severe AE would probably be one, obviously that's worse than mild and moderate in terms of the actual normal reference ranges, and it might be one that might require medical intervention. So I think hopefully that is clear. Thanks.
Thanks very much. Again, let's just alternate. Lynn, a question for you about pediatric extrapolation. Thoughtful question here. For pediatric extrapolation, can you use modeling and simulation before starting your trial for a better informed program?
Yeah, thanks, Leonard. That is a really good question and a very important one. So as I mentioned, extrapolation, this idea of extrapolation is to leverage what we know about a reference population, an adult population or older pediatric population, and apply it to that population that you're interested in studying, which is usually a younger target pediatric population. And the whole notion of modeling and simulation, that is to take data that we have and to understand those relationships within the data using computational methods. That idea of modeling and simulation is a critically important component of the analysis of data. Not just, people say, well, of course we do modeling and simulation to derive a dose, if we want to model a dose in children that could match an adult dose that was effective. Yeah, that's clearly something that we have and that we do in even overall drug development. But one of the things where I want to point out another important place where modeling and simulation can be useful is in evaluating the data that we have that even supports similarity between an adult and pediatric population. So you may have data from different published studies. You may have data from chart, from case studies. You may have data from registry studies, and you may have animal data, for example, that can help be used to support a similarity of the disease between a pediatric and adult population. Well, these types of data and using modeling are tailor-made to understand the relationships. So I think that this is a great way to use modeling and simulation, in addition to the uses once you have established an extrapolation concept and want to figure out dosing, etc., as part of a plan. I'm sorry, I'm losing my voice, but thank you.
Thanks very much, Lynn. Are you up to another question? We'll see how your voice holds up. But this one,
I just took a drink, so I think I'm okay.
Okay, I'm glad you recovered. So here we go. I think this one it's a great question because it opens up a couple of important points for discussion. So what are some ways to address challenges in conducting clinical trials in pediatric patients? For example, how can you obtain sufficient information when the pediatric population available for the study is very small? Any thoughts about that?
I do. And in fact, we convened a workshop 2 years ago on innovative trial designs that specifically could be considered as part of a pediatric drug development program. And by the way, the reason I think that this topic is so relevant, even if you're not interested in the pediatric drug development per se, is that the same principles that apply to developing drugs for small populations also applies to rare diseases. So many pediatric diseases are considered relatively rare compared to the adult population, just like rare diseases overall. So the same principles and the same strategies and the same innovative trial designs that we use in pediatric drug development can be applied in many cases to rare disease drug development. So the idea of Bayesian approaches, the idea of, externally controlled studies, the idea of N=1 trials, all of these potential strategies, and of course the use of modeling and simulation to help decrease the number of patients that you might need. All of these approaches are discussed in the ICH E11A guideline that I described to you and is available now on the website for ICH. So it's really important to consider, and we have many different approaches, and that guideline, I think, really provides excellent examples and references for you. And again, I would encourage you to read that, because if you're interested in rare disease development, some of these same study methodologies and innovations also apply there. Thank you.
Thanks for a great answer. I guess emphasizing that one really needs to be as creative as one can at getting the information that's needed. We're running close to the end of the session. Shabnam, I have one other for you. I think this is regard to safety information. What type of information would go into the investigator's brochure?
Okay, thank you, Leonard, for that question. So the investigator brochure or the IB should have in it all the information that's important for the investigator who's administering the drug to human subjects to really know and understand. And it's required that the information include information on drug substance and formulation and the pharmacological and toxicological effects of the drugs, and then again in humans if known. And then obviously information relating to safety and effectiveness in humans obtained from prior clinical studies, and that's going to be key. Also possible risks and side effects that are anticipated on the basis of prior experience with the drug under investigation, and then also experience that are you can have from related drugs in the same class, for example. And then lastly, it should include precautions or special monitoring to be done as part of the investigational use of the drug.
I think there's another talk on investigational brochures tomorrow as well. Thank you.
Great. Yes, I think some of that will be covered. Just one more for you, Shabnam. Interesting question. In the hypothetical case, does the investigator report death or anaphylaxis or both? I presume if the patient has an anaphylaxis and dies, how does that get reported? Any thoughts about that, Shabnam?
Yeah, thank you for the question. So, the investigator would report to the sponsor this AE as a death, and the cause of death, should be clearly communicated as due to anaphylaxis in this case. Death by itself would not be very informative without letting the sponsor know what they think is the cause of death. And in addition, a causality assessment by the investigator should be provided to the sponsor. As much information about the event as you could note would be helpful to the sponsor, who would then ultimately determine when and whether to report to FDA in this hypothetical example.
Great. Thanks for the answer there. So I think that pretty much brings us to the end of this session. Again, a vote of thanks to both of you for excellent talks and Q&A session addressing safety and addressing specific populations. I think it brings to the close our content for today, which is really focused on clinical trials and what happens in clinical trials. Tomorrow we'll reconvene, and I think the topics that we'll begin with are non-clinical and early clinical drug development, which we haven't discussed yet. They include chemistry and manufacturing controls, pharmacology and toxicology, and clinical pharmacology, all very important topics that obviously we base our design of clinical trials on. Following that, we'll have a couple of lectures, one on ethical considerations, which we haven't touched on yet. I'll be giving a short lecture on innovative trial designs and approaches, and then we'll finish off tomorrow's sessions with two talks on very innovative treatment modalities that are just coming to the fore and offer a lot of promise for diseases that we really couldn't deal with before. So an exciting lineup for tomorrow, and I'm very much hoping that everyone will join us again. And at this point, I'm going to hand over to Brenda, who I believe may have some closing housekeeping announcements.